Hello everyone, welcome to Biology 6 Mark Questions Part 2. There is a Part 1, this is Part 2, just a different type of question. If it's higher only, I will let you know so that you don't waste time doing it if you're foundation. So, one of the most common questions that's asked is about alveoli and gas exchange, or villi and how food moves into the villi. So this next question is about villi, but the same would apply. It wouldn't be the same answers, but the way of doing it is the same for alveoli. Okay, so we've got some villi here, and villi are in the small intestine. Okay, so if we cut the small intestine round like this, then the inside would have, would have many of these little projections here, one of which is this, which is a villi. Now, the food obviously is inside the small intestine here, and it needs to go into the villi. So the food is here, and it needs to get into this good blood supply so it can be taken down around the body. So let's have a look at our question. Our command words on this one, we have got describe and explain. Now, in order to get full marks, we must have some describe and some explain. If they're all described, we can get about a maximum of 3 or 4, because it's actually asked us to do both. So, describe and explain how the villi are adapted to maximise the rate of absorption of the products of digestion. So all that means is, how are they adapted to make sure that enough of this food gets into our villi? Right? So, answer to every biology question, increase in surface area, but it's also giving you another clue here. So not only have we got the villi that increases surface area, we've also got these things called microvilli. So that's given us an extra clue there of what they want. The reason we've got microvilli is to further increase the surface area. That's a description. Okay. We have got a good blood supply here to take all that food away. And if you look here, they've actually drawn this layer here 1 cell thick. So there's a short diffusion pathway, or a short distance that this food has to move to get into the blood supply. So let's have a look at the mark scheme. If you want to pause the video and have a go, that's absolutely fine, do that now. Okay, so mark scheme coming up. Right, so I said there were clues in the question. So if it says D here, just get my pen, where's my pen? If it says D here, that's a describe, you're saying what you see. And if it says EX, that's an explain. So describe many microvilli, and the explain is they've got a large surface area. Right, we've got another describe down here. Again, you can see this on the picture. Many capillaries are a good blood supply. That's our describe. And our explain, to maintain concentration or diffusion gradient or quickly removes food, one of those. Then we've got another describe. Thin wall or 1 cell thick surface, or capillaries near surface. Explain. Short distance for food to travel. And that is 6 marks. Okay. However, if you didn't get those, there is 1 more mark available down here. You can't see this one on the picture. Many mitochondria, and explain, provide energy, or ATP for active uptake or transport. That is like an extra one. You don't need that to get your 6. So to take from this, if it says describe and explain, make sure you've got some description, say what you see, and also some explanations. Otherwise you can't get full marks. Okay, so well done if you got 6 on that one. Let's go to a different type now, different type of question. So we're going to look at some data. Now there's a lot of information on here, so I suggest you pause the video and read this bit on the left hand side, and then we'll have a look at the question. Okay, hopefully you've had a read. So this is all about cholesterol, LDL cholesterol that's in the blood. Now LDL cholesterol comes up in foundation, it also is in combined. However, this question is a level 6. Okay, so you can still listen if you're foundation. Now, LDL cholesterol is found in the blood, and it says over here in the description that the cholesterol may increase risk of heart and circulatory diseases. Now, we need some cholesterol in our bodies because actually cell membranes have cholesterol in them, but we don't need an excess of cholesterol. We don't need too much. So this is all about 4 treatments, A, B, C and D. And they are statins, so they are drugs, a tablet that is supposed to reduce the cholesterol. These are new statins, and this graph is showing us how effective they are. Now, when I first read this, I read it as this was A and the cholesterol was quite low. But that is not what's happening. This is where the drug A was administered, so given to the patients. And then after they had it, the cholesterol actually came up a bit. When they had B, the cholesterol came up loads here. When they had A and B together, the cholesterol dropped quite rapidly there. When they had A and C together, cholesterol came up. And when they had D on its own, it came down. Okay, so our question is, our command word here is compare the effectiveness of the 5 treatments in reducing the risk of heart and circulatory diseases for this person. So it's 4 marks actually, this one, not 6, apologies. So if you look at that graph, you're going to be looking for which drug, when it was given, actually then reduced the cholesterol. So let's just take an example of D. Let me get it on my, right, here we go. D seems to be the best treatment. It's not asking us to say why. It's not asking us to provide any numbers. It's just asking us to compare the effectiveness. So we're looking for ones where it went down. So actually A and B was quite effective, wasn't it? When they had A and B. We'll do the mark scheme in a minute. I'm just jotting down ideas at the moment. A. Was A effective at all? No, because it's put the LDL cholesterol up. So A on its own, not effective. Okay. B on its own, not effective. A and C not effective. Have we written that one? No, not yet. A and C not effective. Now, what about C on its own? We don't have C on its own. So, no information about C or, oops. So, or we can't say whether C is effective. Okay, so we're just looking at that graph and, excuse me, got an itch, and seeing which one reduced the cholesterol. So let's have a look at the actual mark scheme. Okay, I'll rub out those red bits so you can see it properly. There we go. Right, so first one, A and B most effective. D is the best single treatment, or D is the most effective there. Neither A or B alone are effective. And cannot tell if C is effective, because we don't have any information about C. Okay, so if you need to pause the video and jot any of those down, that's absolutely fine. If not, we're going to go on to the next one. Okay, evaluating stem cells. So this is quite a tricky one. Again, stem cells come up in foundation. You wouldn't have to evaluate in foundation, but you may have to talk about them, so it's worth listening to the knowledge. So the first thing is, what are embryonic stem cells? Okay, so let's have a look here. Where is it? Here. So embryonic stem cells. So embryonic stem cells are when a sperm and an egg have just met and fertilized, and they divide. This is an early embryo. Okay. Now these are embryonic stem cells, which means all the cells are identical, because a stem cell is an undifferentiated cell. Now you might think, well, where do I get these embryos from? That's odd. You know, having embryos just in a petri dish in the lab. Well, these are often embryos that are left over from fertility treatment, and the parents don't need them, so they might be donated as stem cells, or sometimes if it's legal they can make these in a lab, but more often than not they're from IVF treatment and they're left over. But obviously they have the potential of growing into a baby, so that's why there's some ethical things around this. So let's have a look at the question. Let's go back to the question. Evaluate the advantages and disadvantages of using embryonic stem cells as a potential treatment for diabetes. Include a short conclusion. Now, whenever we evaluate, we must put a little conclusion, because if we don't, we can only get half marks usually. Even if that conclusion is, I agree with using them or I don't agree with using them, you have to put a conclusion. Right, so now we know what embryos are, embryo stem cells are, and the fact it's evaluate. Let's have a look at what we could put in there. Okay, so we've got lots of writing here, so let's just go through it a little bit at a time. Advantages of using embryonic stem cells to treat diabetes. They differentiate into many types of cell. So those embryos, scientists can actually make them turn into any type of cell they want. So they might want a heart cell or a skin cell or a nerve cell or a muscle cell or a heart cells. Now the posh word for that they can make all types of cell is that one there, pluripotent. Okay, pluripotent. So we could turn them into pancreas cells and help them with the damaged pancreas. So there is a low level risk of rejection. That's because they are coming from embryo cells. They're less likely to be rejected. Whereas if you transplant a pancreas, it's more, you'll have to take anti-rejection drugs forever. And if you've got pancreas that is working, then you don't have to take insulin anymore. And you don't have to wait for a donor, because obviously for you to get a donor, usually someone has to die. So that's not a nice thought, is it? Right, let's have a look at the disadvantages. Some stem cells can get mutations in them, which is where the DNA slightly changes. So they could become cancerous. Then we've got an unethical bit here. Now, if you're going to ever say anything is unethical or morally wrong, be specific. Okay? So in this case, it's unethical because unborn babies are being killed. If it was, let's say it was a question about animals, you could say, this is unethical because you can't ask the cow if it wants to do whatever. Right, and the other thing is, yeah, it's quite expensive to do this. So there, we've got some pros, some good things, some advantages, some disadvantages, and then just let me move my picture across the screen. So, and then you must have your conclusion, okay? Overall, now it doesn't matter what your conclusion is. You can agree, you can disagree, as long as you say something. So overall, I think stem cells are a good way to treat diabetes, as they're not going to be rejected and are more convenient than insulin. Okay, so just one little line of conclusion. You could even say, I agree with using stem cells because of the reasons above, but you have to have that conclusion there if it's evaluate. Okay, well done on that. Ooh. So 3 advantages, 3 disadvantages, and a conclusion. That should give you full marks. Right, let's have a let's have a little look at this one. So this is stem cells used to treat some human diseases. So what I suggest you do, just pause the video, have a little look at the information there, have a little read. Okay, do that now. Right, so this one takes a slightly different tact. This talks about stem cells from early embryos, so the stem cells like we talked about before. But it also talks about stem cells that can be collected from adult bone marrow. Now for this, it says it's simple operation but may be painful. They actually drill into your thigh bone to get these, but then if you need some new cells for something, you know, a bit of pain. But on these, the only type of cells that you can get from adult stem cells are blood cells. So we can't make all the other types of cell, just blood cells. So we can treat blood disease. And then recently there have been trials of other types of stem cell from bone marrow, and these are used to treat diseases such as heart disease. Okay, so given us all this information, what do they want us to do? They want us to evaluate the use of stem cells from embryos or from adult bone marrow for treating human diseases. Okay, and again, you should give a conclusion. It doesn't always say give a conclusion, but if you see the word evaluate, you need to give one, okay? Right, if you want to pause the video and have a go at this, do now, please. So let's have a quick look at the mark scheme then. There's a lot on this, but you only need 6. Let me get rid of my scribblings. Will it? Yeah. Okay. So we've got, now you only need 6 of these. 3 from this top bit here. So 3 for this one. 3 for this one. And this could be 2 pros and 1 con, or it could be 1 con and 2 pros, as long as we've got 6 altogether. So embryo stem cells can treat a wide variety of problems. Many available. Using them is better than wasting them. Cons, bad things: harm or death to embryo, untested, unreliable, may not work, do not know the long-term effects, Embryo can't be asked if it wants to be used. And then adult bone marrow. So if you're using your own bone marrow for stem cells, there's no ethical issue. You can give them yourself and give consent. Relatively safe. Does not kill the donor. It's low risk. We know how it works. There's been lots of tests. Cons for adult bone marrow: operation hazard, so just risk of infection, things like that from operation. We don't get many types of cells, so not many problems can be treated. And it is quite painful, so it may deter donors. If you're not using your own stem cells, you might be using somebody else's. And then we just need a little conclusion, and then that would give us 6 marks. So in these 6 mark questions, we don't have to write loads. We don't have to write essays. You can even bullet point them if you want. It's absolutely fine. I find it easier to do that. So hopefully that's been helpful. And I will see you in the next video. Take care, everybody.